What we're going to do in this lecture is pull together what we've learned about the cardiovascular system and think about it in the context of how it's controlled. So we know the cardiovascular system is a very fundamental, important organ system in the body. With our focus on control, we're basically going to revisit how the heart works and how blood flows through the vessels in the context of control. So the question here is, how is the cardiovascular system controlled? And a lot of our focus is on the cardiac output. The cardiac output is how much blood emerges from the heart per unit time. And since the circulatory system is effectively a closed system, that is, its volume from moment to moment doesn't change very much, then that cardiac output is actually the flow rate at any position in the circulatory system. The cardiac output is quite simply the heart rate, the beats per minute, times the stroke volume, how much the volume of blood that emerges from the left ventricle. Now, that's going to be our focus in terms of the efferent side of our control system, how the heart is varied, the role played by the blood vessels. And ultimately, we want to relate that output to the blood pressure, which is the state variable for the control system that we're going to consider. Now, blood pressure, as you know, it's always measured when you go to the doctor's office. It's really important as a physiological metric of health because when it's too low, hypotension, you have a variety of catastrophic effects. And this illustrates how multiple organ systems and really all the cells of the body are so dependent on the cardiovascular system. If you have too little cardiac output and too low a blood pressure, then you have insufficient transport of nutrients and dissolved oxygen, which will result in fatigue. Ultimately, you can have shock or organ failure if you have sustained chronic hypotension. Now, hypertension is a really big problem, especially given our American diets. Too much pressure in the circulatory system can strain the contractile cells and the connective tissue, ultimately leading in heart failure. You can have aneurysms, which is a bursting of blood vessels. The kidney, which depends on hydrostatic pressure, can be damaged when those pressures run too high. And ultimately, you can cause a stroke, an aneurysm in your brain that leads to real challenges. So, blood pressure is a fantastic state variable for a negative feedback system that maintains homeostasis on the circulatory system. So first we're going to consider cardiac output, and then we're going to see how that relates to blood pressure. Now, what's fun here is we can revisit all the topics that we just covered on the cardiovascular system in this context of control. So, for example, for the control of muscle activity, we talked about how the SA node has autorhythmic cells that generates the pacemaker potential, generates the action potentials that drive the contraction of the heart. So those autorhythmic cells in the SA node, which set the pace for the whole heart, the AV node essentially follows along the lead set by the SA node. Those autorhythmic cells are innervated with autonomic nerves. They have both sympathetic and parasympathetic nerves. And they have beta-1 receptors, which makes them responsive to the adrenal sympathetic pathway. Now let's monitor the autorhythmic cells in the SA node to consider how it's influenced by these inputs through the autonomic nervous system. Here we have the membrane potential of one of those autorhythmic cells. And this is what we've seen before. We have that depolarization by the cells themselves. This is just an inherent property of the autorhythmic cells. and that causes this rise in membrane potential. This is the sort of last little bit of an action potential. So as the pacemaker potential rises, it reaches threshold, and that initiates an action potential. And we know now that the action potential is fixed in its duration. But the pacemaker potential can be influenced by the autonomic nervous system. So for example, when you become more active or stressed, then your sympathetic pathways become recruited, and the release of norepinephrine through the sympathetic pathway causes a shortening in the pacemaker potential. So even though the action potentials won't change in duration, and you can see the two action potential curves just lay on right on top of each other, that's fixed. But the duration of the pacemaker potential is flexible. When you're stressed out, sympathetic pathway, then there's a shortening of that, which is going to increase the beat frequency. Now let's start to lay out our control system. And again, we're looking entirely on the efferent pathway here. Everything I've told you about the cardiovascular system, you can think of as being part of an efferent pathway in control. We haven't said anything about the sensory or afferent side of our control system. So we have this release of norepinephrine by neurons along the sympathetic pathway. The beta-1 receptors in the autorhythmic cells of the SA node are responsive to norepinephrine. This shortening of the pacemaker potential is driven by actually an increase in the duration of the IF channel. So the IF channel effectively becomes more active. It causes a shortening of the previous action potential and drives the membrane to threshold at an earlier point in time in the cardiac cycle. So as a consequence of that, you have the increase in heart rate. Now, what I've overlaid here is what occurs to the pacemaker potential when it's exposed to acetylcholine. The acetylcholine is released by the parasympathetic pathway. So this is what occurs when you are relaxed, and you can see that there's an increase in the duration of the pacemaker potential. So we're spacing out the action potentials in time. In other words, we're reducing the frequency of heart beating. So we have that as an efferent pathway, the parasympathetic efferent pathway, where we release acetylcholine onto the SA node. That causes an increase in the permeability for potassium. Okay, so we got to think back to the neuron. Increase in potassium permeability, that's going to hyperpolarize the membrane, right? We decrease the permeability of the membrane to calcium. That's going to do the same thing, hyperpolarize the membrane, which doesn't influence the action potential, but it does cause the increase in the pacemaker potential because we're effectively hyperpolarizing the membrane and making it so that it takes longer to reach threshold. Okay, what about stroke volume? So I've said this is all about heart rate. The cardiac output is the product of heart rate and stroke volume. So let's consider stroke volume. Now, stroke volume I first mentioned when I described this principle, the Frank-Starling law. And if you'll remember, this is the relationship between how much blood you get out of the heart, that's the stroke volume, as a function of how much enters into the heart, in particular the left ventricle, when it's relaxed. And fortunately, this is a positive relationship. So you get more blood out as you pump more blood in. And this is the curve that you would expect in a relaxed state. But when you become more active or more stressed, what happens is that this curve changes. And it has a more positive slope. And so this is what we see in the presence of norepinephrine, our sympathetic pathway. And so the resting value here is much greater. So in other words, you get more blood out, a greater stroke volume, for the same amount of blood into the system. That means less blood is left behind at the end of systole. This is achieved by increasing the force generated during contraction. Now, norepinephrine actually has two effects on the heart. So in those myocardial cells that have beta-1 receptors, first of all, so by the way, this is the contractile cells, not the autorhythmic cells. Thus far on our control system, we're only talking about autorhythmic cells. Now we're talking about an effect on the contractile cells. So this is a sympathetic pathway acting on those contractile cells. So when they're exposed to norepinephrine, there's an increase in the concentration of calcium that is released by the sarcoplasmic reticulum when it's activated. Now that increase in intracellular calcium is going to result in a greater twitch tension. I've mentioned before that it's possible to get a greater twitch tension only in cardiac muscle. Here we see what causes that, norepinephrine. The other thing that happens is that there's a shortening of the duration of activation. So the twitch duration is shorter. All right, so those two factors are what we call contractility. Okay, when you have in the presence of norepinephrine an enhancement in contractility, which means you're going to get greater twitch tension through more calcium and a shorter twitch duration. So that increase in contractility results in a higher stroke volume, which is exactly what we see in this modified version of the Frank-Starling law. Okay, so when we now see that we've got an increase in cardiac output through these two pathways. One, this contractility is through the contractile cells. The other two pathways act on the autorhythmic cells. Now let's return to our conceptual model for the blood pressure in the major arteries. Now, if you'll recall, we have a plunger here, which models the pressures generated by the left ventricle, the semilunar valve. This water balloon-like structure is modeling the aorta and the major arteries. And then we have the vessels that branch off from that. So cardiac output, again, is the product of heart rate and stroke volume. If you have a low cardiac output, then either through heart rate or stroke volume, you're going to have less blood entering into the major arteries per unit time. So again, this is a trace of pressure up top, where we can take the temporal mean, that's known as the mean arterial pressure. So this is what the curve would look like if you have a relatively low cardiac output. If we increase, say, the stroke volume through the contractility of the contractile cells, now we're going to be injecting more blood with each contraction, and that's going to serve to elevate all pressures, including, or therefore, it's going to elevate the mean arterial pressure. So what this illustrates is how cardiac output relates to blood pressure. All of these factors that we've mapped out on our efferent pathways have an influence under blood pressure because they influence the cardiac output. Another factor that influences the blood pressure comes from the peripheral resistance. So we've established before how the diameter of the arterioles, which we're modeling here very simplistically just as a single vessel, but of course there's thousands of them, the diameter of the arterioles in the body influences, because of Poussey's law, the amount of peripheral resistance that there is. So that's going to effectively control the output of blood as it flows from the major arteries. So if you have vasoconstriction and a reduction in the diameter of those vessels, that serves to maintain blood volume throughout diastole. So if we look up top at how pressure varies over time, in this case, the green curve illustrates what occurs under those conditions. You have an elevated diastolic pressure, which in turn causes an elevation in the mean arterial pressure. So if we turn our attention back to our control system, we can add that. We've got alpha receptors in the arterioles, which respond to the release of norepinephrine by contracting That vasoconstriction causes a reduction in diameter and a large increase in peripheral resistance. Okay, that's going to tend to increase blood pressure. So all of these sympathetic responses lead to increases in blood pressure. All of the parasympathetic responses lead to a reduction in blood pressure. Now, one thing I'll say about the peripheral resistance is that when the sympathetic pathway is inhibited and there's a reduction in norepinephrine, then that triggers a vasodilation. So there is no parasympathetic input to the arterioles. Instead, a mere reversal of this pathway will result in vasodilation. And that's because the sympathetic neuron has a tonic level or a constant level of action potential frequency. And so when the sympathetic pathway is inhibited, there's a reduction in that frequency, which results in less release of norepinephrine. What about sensing? Okay, we've mapped out a very complex pattern on the efferent side. What about the afferent side? Where in your body should you monitor to know whether or not your heart is doing enough and whether or not the peripheral resistance is adequate in order to maintain your blood pressure? Okay, blood pressure is our state variable in this control system. Well, it makes sense that you'd want to pay attention to where the pressures are high. And that's in fact where we find the receptors that matter for cardiovascular control. They're found in the aorta, as you might expect, but also in the carotid artery, the major artery that sends blood to your brain. In those two positions, we have the receptors for our control system. They're called baroreceptors. You can think of the baroreceptors as functioning very similar to the receptors that I described in the urinary bladder, those stretch-sensitive receptors where the dendrites are essentially woven into the connective tissue. And as they are put in tension, those dendrites, then there's a change in the membrane potential of those receptors. Baroreceptors are also mechanically sensitive, and they're woven into the walls of the carotid artery and the aorta. So as it expands, it being an elastic structure means as the baroreceptors are stretched further, that's indicating a larger pressure. And the baroreceptors integrate pressure changes over time, so you can think of them as effectively mean arterial pressure sensors that work by strain in the major arteries. So the baroreceptors provide the sensory means for control. What about the integration center? Well, this is a case where a region of the brainstem controls physiological function. And so we have a negative feedback system, and so it has all of those parts that I described in an earlier lecture on control. That integration center that we have for our control system is found in the medulla oblongata. Okay, that includes our cardiovascular control system. And the algorithm that governs this integration center is what you would expect for a negative feedback system. Okay, so this looks complicated. I've presented it before, but it's the difference between the state variable, okay, that's the blood pressure in this case, that's reported from the baroreceptor. The difference between the state variable and the set point, that is the body's desired blood pressure, when that error or difference is less than a threshold or a minus value for the threshold, that is basically when the blood pressure is below an acceptable level, then the sympathetic pathways are recruited. Whereas if our state variable is much greater than the set point, above that same threshold, tau, then the parasympathetic pathways are recruited. So let's just consider those individually. In the case of the sympathetic pathway, that's triggered by low blood pressure. So that's what I've added to our flow chart. We've got low blood pressure down here. The integration center is going to respond to that provided the error is less than some threshold. That results in an inhibition of the parasympathetic pathway and an activation of the sympathetic pathway. Makes sense. On the other hand, if the blood pressure is too high, and we see that down below for our state variable, then we have essentially the reverse. The sympathetic pathway is inhibited, the parasympathetic pathway is activated, and as I mentioned, you're going to have vasodilation resulting from the fact that you have a reduction in norepinephrine release. Now, another big factor that we haven't yet talked about on the efferent side of things is blood volume. If the whole system has more blood, then it's going to be more inflated when it enters the major arteries. So we return back to our model here. And what we see is the mean arterial pressure in this case, but what affects blood volume? Well, for one, the GI tract, if you drink more water, that water is, unless you're producing diarrhea, that water gets absorbed. Okay, you swallow it, it ends up in your blood. So that's going to be adding to blood volume, which in turn is going to enhance blood pressure. Now, on the outflow side of blood volume, we have the urinary system. So your kidneys can produce more dilute urine, which means that you're going to urinate more and your urine is going to be more clear. And so we have both the kidneys and the GI tract, and for that matter, your behavior influencing blood pressure. So if the blood volume goes up, then if we look at our pressure trace, we would expect both the systolic and diastolic pressures to increase. So an increase in blood volume is going to increase the mean arterial pressure. So we can plug those components into our control system. Let's first consider the high blood volume case, which causes an increase in blood pressure. All right, so as we've mentioned, the sympathetic pathway is going to be inhibited, but now we have an additional pathway, which is we're going to want to, since the blood pressure is high, we're going to tend to want to reduce the blood volume. And we can do that by basically urinating more. So the kidneys are recruited in this effort. You urinate more when the kidneys are trying to reduce the concentration of urine. Okay, so the urine becomes more dilute, meaning you've got more clear urine. That is going to reduce the blood volume because we're allowing more water to leave the circulatory system and enter into the urinary bladder. So in this way, urine concentration influences blood pressure. What about the opposite? That is a low blood pressure situation. For that, the parasympathetic pathway is inhibited. The kidney is actually recruited to enhance the concentration of urine, but you also tend to become thirsty under this condition. Okay, and thirst will drive you to use your musculoskeletal system and behavior to seek out water and to drink it. And once that is absorbed, then you're going to have an increase in blood volume. Now, the kidney and this thirst response are sort of more long timescale actions compared to the sympathetic and parasympathetic pathways, which can be recruited quite quickly in response to changes in blood pressure. Instead, urine production and behavior are a little bit slower and also more long-lasting in their influence on blood pressure. At any rate, what we see here is how drinking water can effectively elevate the blood pressure. So this lecture set out to address the question of how the cardiovascular system is controlled, and this is the answer. And this is what we're going to do when we consider these physiological control systems, is essentially map out all of the parts that constitute the control system and consider how they respond to different perturbations to the system. In this case, an elevation in blood pressure or a depression in blood pressure.